Good morning or afternoon, class. I'm going to ask you to open up another window so then you can watch this video and go through the slides. If you already left it open from the last video, then great. If not, go to the Google Classroom. At the top there is a tab for classwork. Click on that, and then along the left side there is the different units numbered 1 through 10. Go to number 6, which is DNA, RNA, and then go through that menu of items, and at the very bottom of it should be the slides for this unit. Once you get into the slides for this unit, I am on slide 26. You might want to pause the video and try to find that. I'm on slide 26. Alright, on slide 26 there is the title, Using DNA Protein Synthesis. This is a video on transcription and translation. So how do we make proteins? How do we make proteins for hair, your fingernails, your enzymes to break things down? Like for example, one of my favorite enzymes is lactase, which breaks down lactose sugar. It helps you break down the sugars that are found in milk and in dairy. And that's just one of many different enzymes and one of many different proteins. We are full of different proteins. So how in the world does the body make proteins? And how come I can't make proteins that some other organisms can make? Well, it happens to do with your genes. If you had the gene for it, then your body would have the mechanisms to make it, but you need the gene. So if I was to insert the gene into an organism, like for example our GMOs or genetically modified organisms, then they already have the machinery to make the product. They just need the instructions. So that making from those instructions, that's what we're focusing on. The very first step is called transcription. Alright, imagine that you go to a library. Like for example, one of my favorite libraries is in Columbia. It's called the South Carolina Library. And you cannot check out any of their books. You can go in there and you can read and you can look at all their items, but you can't check out a single thing because a lot of what they have is one of a kind. They specialize in things of South Carolina. So they have books from South Carolina people. They have newspapers from the 1800s from South Carolina cities. Even Sumter has a few editions stored there in the library. They also have journals and diaries from people from way back to the colonial days where there is no other copy. There's only one, the one that that person wrote down in that book. So you can't check it out, but you can sit there at the table and you can thumb through. But what you can do, if you need those words, is take a notebook in there and copy it. Open up and read what you want copied and copy it into your notebook. And I've done that several times. This is like transcription. We're inside the nucleus of the library of the cell where the instructions are kept. And we don't want those instructions to leave. We just need a copy of a section of it. So we open up the DNA where the gene is, and we make a copy of that section, those instructions for making a specific protein. This process is called transcription. You probably know scribe or transcript. Script is in there referring to words or instructions. So what we're going to do is we're going to copy the instructions. At the bottom of that slide is a picture of it. And the blue molecule is the DNA, and the red is the RNA. If you look very closely, you'll see that there's the bonds. The A's and T's are bonded together. The G's and C's are bonded together. But you may notice in the red molecule there's no T's, there's U's in every place where there should be a T. A and T normally bond, but whenever we make an RNA molecule, we're going to substitute the T, thymine, for uracil. So every time T is needed, use U instead. That's the exception of the rule. There's 3 rules. A's and T's bond together, G's and C's bond together, and with RNA, every T becomes a U. Now you've kind of more or less mastered the language of DNA in 3 rules. On the next slide, number 27, the nucleotides of RNA bond together to form a single strand. based off of the instructions of the DNA. And this section of RNA that we created from the DNA has a specific name because RNA comes in different forms. This one is called mRNA or messenger RNA. That messenger RNA has the message, has the code, the instructions for doing something. Before that mRNA can leave the nucleus, we've got to do some editing because there's a bunch of junk in our DNA. Things from mutations that the cells realize is a problem, it needs to ignore it, can't get rid of it, but it marked it as don't mess with this. Or viral DNA, viruses that have made you sick, either you personally or our ancestors, is stuck in our DNA. We have a lot of viral DNA in our chromosomes, and they've been marked as don't read this anymore. We can't get rid of it, but let's not use it. Let's not make anything from it. So in the editing process, we're going to use some enzymes. Those enzymes are going to cut away at the mRNA to leave the good information and get rid of the bad information, the junk. What is the junk is going to get recycled, and that's called introns. The introns stay inside the nucleus and can get recycled again, can get broken apart and reformed into new mRNA. What leaves the nucleus is called exons, or the mRNA. Is now going to exit the cell. It's going to be expressed. After leaving the nucleus, we want to protect that mRNA because it's fragile. It can easily be broken down by radiation, can be broken down by enzymes, can be broken down by anything too acidic or too alkaline. So it's super sensitive. And we want to wrap it up in a coat. So a bunch of A's, a bunch of adenines, those nucleotides, are going to get attached to an end. 1,000 or more are going to get attached, and we call that the poly A tail. Poly means many. A refers to the adenine, and it forms a tail that will wrap around it. Then that strand will leave the nucleus. That was all transcription, but let me get the whiteboard just to make sure we're on the same page. All right, so what just happened in transcription? Number one, we took the DNA and we made it into mRNA. The DNA had a gene, a set of code, and we just copied that single gene. We didn't copy the whole strand of DNA, the whole chromosome, just a section of it. Where this happened? In the nucleus. That's where the DNA is stored, so we copied it in the nucleus. We're going to make the DNA into mRNA, and there is a rule with this. Hopefully you can make out these letters in red. The A's, T's, G's, and C's are going to get transcribed. The A's want to bond with T's, but we don't use T in our RNA, so we're going to substitute every T for a U. If you can read that, that's a U for uracil. T's want to bond with A's, that stays the same. G's bonds with C's, and C's bond with G's. And this is going to be our bonding rule. This is the DNA. When we open it up by enzyme, an enzyme then added all these together to bond with it. So if our code had an A, we had a U, a T, an A, a G, a C, all the way down. This is our new code for mRNA. The next stage is called translation. Transcription just made a copy of the instructions. A copy is not useful until you do something with it. So what we need to do is we need to translate it into a thing. And this is going to be a protein. We're going to make an enzyme or a channel or a pump or some sort of structure that the cell needs. Where is this going to happen? Well, we left the nucleus. We're now looking for a ribosome. And the ribosomes could be free floating out in the cytoplasm of the cell, or can actually be attached to the endoplasmic reticulum. Wherever it can find a ribosome, the mRNA is going to feed through the ribosome and it's going to start making our protein. But it's got to read it in a code. All these letters that we made in transcription get read 3 letters at a time. So the first one was a U, followed by an A, followed by a C. That's 3 letters, U, A, C. That's our first codon. The ribosome is going to read that codon And that codon is instructions for making tyrosine. Tyrosine is amino acid. You may remember that amino acids are our building blocks for proteins. So that's just one amino acid. What's the next one? GUG is the next one, and that's going to be for the amino acid valine. Now, that was only 2 amino acids. In our gene, there's probably instructions for making protein with 100 to 200, 500, or 1,000 amino acids all chained together. So it's going to keep adding one amino acid after another amino acid after another amino acid, and all those together are like adding bricks in a line, one brick behind another, and you keep adding them. Eventually you have a building or a house. But what we need to do is start with one brick at a time, and not all these bricks are the same. Some of them are a little bit bigger, some of them are a little bit misshapen compared to the rest, and that's going to be very important because we want to make a molecule that has a distinct shape. You may remember the Pac-Man, the enzyme. It has the active site, and that active site has to have the right shape for another molecule to fit inside of it. If it has the wrong shape, then it can't do its job. So all those amino acids have to be paired up in just the right way to create the right shape for the active site of an enzyme. Or if we're talking about a channel or pump, it's got to be shaped just the right way to allow the molecules to either be pumped into or out of the cell, or a channel to allow it just to facilitatively flow through with some sort of passive diffusion. Now, there is some steps involved with figuring out what amino acid we need. I'll pull up the whiteboard one more time. So, originally we had DNA, then we went to RNA, specifically mRNA, and then we paired them into threes, one set of 3 at a time, and that's called a codon. Codons. So here's codon 1, here's codon 2, and then the codons tell us which amino acid. Now, some brilliant scientists have spent time trying to figure out this code of life. And it turns out that all living things, from bacteria to plants to animals, you and I, we all use the same instructions. So if we compare the code from a bacteria to you, if we had the same 3-letter code for one codon, it would say for the same amino acid in the bacteria as it does for you and I, or a plant, or anything else. It's the same instructions. We just need same code. It's just the differences in the instructions as to how to put those codes together to make a different protein. All right, I'm gonna hold this up for you. Hopefully the layer's not too bad. This is a codon chart. And the codon chart tells us what amino acid is used for the code. So this codon chart is read using those 3 letters. On the far left is our first letter that we use, either U, C, A, or G. And you may notice that it says U because we're dealing with RNA. Along the top will be the second letter, and then the far right will be our third letter. And then inside these boxes are the different amino acids with their codons. This is what scientists spend a long time trying to figure out for the code of life. If you want to see what that looks like a little bit better or more clear, then you can go to slide number 31. So slide 31, we have the first letter on the left, the second letter on the top, and the third letter on the right. And let's say the codon, I have an example down there. The DNA sequence was originally GAC, G-A-C. Well, we need to transcribe that first. The G wants to bond with C, so first letter is C. A is the second letter, but A wants to bond with T, but we don't use T in RNA, so we're going to use U. And the third letter is C. C is going to want to bond with G. So our 3 letters are going to be C, U, G. That's our RNA code, CUG. CUG if we were to use the codon chart above and first start on the left, we find the C, and then we go along the top and find the G and see where those two crisscross. The C and U's leave us 4 choices, and they're all Laocene. But let's say that there was more than that one choice. Then we need to go to the far right. Far right, look for the G, where those 3 letters crisscross, the C, the U, and the G, they crisscross with Laocene. Laocene is amino acid, and all these on this codon chart are amino acids, except you may see there is some exceptions. There's 4. AUG is the codon, AUG. If you want to pause the video briefly to try to find it, AUG. All right, it says start. That's the start codon. So sometimes there's junk in our genetic code, and we want to, the ribosome is going to read the code a little by a little, until it gets to AUG, and then it will start making our protein. So that's the starting point. Kind of like when you are reading a book, you don't just start in the middle of a sentence, you start at the beginning of the sentence. So sometimes there's stuff before it. Well, it's going to ignore the stuff before it and only work at AUG. And I remember that school starts in August. That's our starting point for all organisms. Then you may notice that there are some stop codons in the top right of the box. UAA is a stop codon. UAG is a stop codon. And the last one is UGA. Those 3 stop codons tell the ribosome when to stop adding amino acids. Because remember, there's a poly A tail with a bunch of A's, 100 or more, attached at the end of the molecule. We don't want to translate that. So there's going to be a stop codon that says, don't do any more at the end of this. Ironically enough, and this is just irony, in the English language, there is 3 things that tell us when we are done with a sentence. Do you remember what they are? A period, an exclamation point, and a question mark. In the codon world of our DNA, there is also 3 things to tell us when to stop. 3 different codons. Just a coincidence, wasn't planned that way. On the next slide, which is number 32, there is some practice. There's 3 different examples of some DNA sequence. Remember, you first got to switch it from DNA to RNA. So I'll help you with the first one. TGC is the DNA molecule. And we need to turn it into RNA. So T will become an A, G will become a C, and C becomes a G. Remember, A's and T's bond together, G's and C's bond together. So the codon for the first one will be A, C, G. And then you can use the codon chart above to figure out what amino acid would go in the sequence. And you can do that with all 3. On slide number 33, the next slide, this is a proposed new codon chart. I just have it in slideshow just in case you ever encounter it. But most textbooks, most professors, most sources don't use this yet. It's still somewhat new. What you do is you start in the middle and you work your way out. Think of it as a bullseye, but you start from the inside and you work your way to the edge. So the very first letter is, I have on the right, UUC. You can start with a U in the middle and then find the next U that's still in that quarter of the circle, and then find the C that is on the outer loop, and then you have your amino acid. This is much faster than the other version of a chart, but again, I need you to know how to use the other one. I wanted to show you this one just in case you encounter it. Alright, slide number 34. The sequence of mRNA nucleotides determines the order of amino acids. So those nucleotides, if it's an A there, it's going to be very different for a product than if it was a G or a C or a U. So the order of the nucleotides dictates what amino acid is going to go where, and then those amino acids are going to dictate what protein we have. And that's what the instructions there, or on the slide, say. That image, if you're looking at it, is not translation, it's transcription. We have the DNA molecule and we got the mRNA there being formed. A very short section, that's not very realistic, but it gets the idea. All right, I have a couple more slides that I want to introduce you to. So the next one is number 35. I've introduced you to mRNA. mRNA, the M stands for messenger. It has the instructions, but that's not the only type of RNA that works with transcription translation. There's also tRNA. The T stands for transfer. What is it transferring? It's transferring the amino acids. Those amino acids don't just poof into existence. They got to be brought to the ribosome. The ribosome only puts amino acids together, one behind the other. Well, it needs those supplies. It needs those amino acids, and tRNA attaches to amino acids and brings it to the ribosome. And the tRNA has on it a code called an anticodon. That will make more sense on the next slide, which is slide number 36. That bar that is in purple, it has a start codon and stop codon at the two ends, and it has the codons broken up into threes. The parts that look like clovers, something like that, has some code at the bottom of it. That code is the anticodon. It matches up with the codons. The G's and C's are bonding and the A's and U's are bonding. If the tRNA matches the mRNA, then the amino acid will be left with the ribosome to be attached to the next amino acid and the next one, the next one, the next one to make our protein. If you're a little confused as to what AA stands for in those circles and a square and a hexagon, octagon, I'm not sure what shape that is, how many sides that is. Hexagon is 6-sided. Those are amino acids. AA is amino acid, and they have a number. All of them were numbered. So that's why you see a number with them, 6, 12, and 7. The next slide is 34, same thing, same idea, with the ribosome shown in black. The red is the tRNA, and then the amino acids are shown in green bubbles, and you see the codons at the bottom. All right, so there's mRNA that has the message. There's tRNA that holds the amino acids and brings it to the ribosomes. But then there is one more form of RNA that I have skipped. How did I miss it? That form of RNA is actually on slide 29. I had to go backwards a little bit. The third form of RNA is called rRNA, or ribosomal RNA. The ribosome actually has two pieces stuck together. So two different pieces of a molecule. And what's holding those two pieces together is the rRNA, the ribosomal RNA. Without the rRNA, the ribosome falls apart into two separate units. That's the purpose of the third type of RNA. Ribosomal RNA holds the ribosome together. The mRNA, messenger RNA, holds the message instructions for making our protein. And the last one is our tRNA, which transfers the amino acids one at a time to the ribosome. I hope this helps. I'm going to post an assignment related to transcription translation as soon as I can. And I'll also post a copy of the codon chart so you can see it, and a couple of videos that walk you through this process of transcription translation. Hope you have a great rest of your day.